Hello. I came to COA because I wanted to study field biology, but within that area, my interests have been pretty eclectic. I'm just generally fascinated by living things, so that's why my senior project has 3 parts. I wrapped up 2 different research projects I've been working on over the past couple of years, and also did another project in a newer area of interest. Part 1. 2 summers ago, I fulfilled my internship requirement by participating in a research experiences for undergraduates program. I spent that summer in New Hampshire with another student designing and carrying out a study of salamander ecology, basically spending a lot of time in the woods rolling over logs looking for little critters like this red-backed salamander to find out which species live where in relation to streams and each other. Some herpetologists have suggested that monitoring salamander populations might be a good way to monitor environmental health, so we thought it would be useful to add to the body of knowledge about what salamander distributions look like under relatively undisturbed circumstances. The winter after that, I spent an independent study writing up our results in the format of a scientific paper. Then this fall, I brought it to the advanced research tutorial and revised it based on my professors' and classmates' comments. The ultimate goal has been to submit that paper for publication so that our results might get out there and be useful to someone, and it still needed revision and polishing to make it as good as it can be. So as part of my senior project, I worked through 2 more drafts, making 8 drafts total, and I feel a lot happier with the way it presents, analyzes, and discusses our study. This study and experience has been kind of hanging around in the back of my mind since that summer, and I'm glad my senior project gave me a chance to put some time toward wrapping it up. Part 2. This past most recent summer, I did another research experiences for undergraduates program, this time studying pollination at the University of Massachusetts Amherst. This study was about how pollinators, like the squash bee in the middle of this picture, and herbivores, like the striped cucumber beetle towards the bottom, sense and locate squash flowers. This experience was important to me because it gave me a chance to conduct a field study with potential agricultural applications, which is something I've been becoming increasingly interested in. As the second part of my senior project, I wrote up the results of this study and presented it as a poster at the Maine Biological and Medical Sciences Symposium at MDIBL. Part 3. The final third of my project is about these plants, swallowwort or dog strangling vine, which originated in Eastern Europe and has 2 species that are invasive in the northern United States and southern Canada. Black swallowwort, which is pictured in the upper corner, and pale swallowwort, which has lighter flowers. They're both perennial, wind-dispersed vines related to milkweed, and they form dense mats, as shown in the lower picture, which exclude native flora and other vegetation. For the third part of my senior project, I used information provided by researchers at Cornell, looked up more information on online herbarium databases, and mapped as much of the species' current known range as I could, using Geographic Information Systems or GIS software. I'd taken 2 GIS-focused courses while at COA, and I was really excited to explore some more of what can be done with this technology. In this map, the black dots represent known locations of black swallowwort, and the yellow dots represent pale swallowwort. From those current locations, I wanted to project where the plants might spread to, and for this I got to use some of the really fun capabilities of GIS software. This picture shows one of the models I set up using the Gis Modeling tool, which looks cool once you get it set up right, and is actually a lot like playing with dominoes because you basically just set up a series of commands that tells the software to do certain things in succession, and then you hit go, and then you find out if you did it right. So I got GIS maps of climate variables like mean annual temperature from the National Oceanic and Atmospheric Administration's website, and used the modeling tools in the GIS software to create a map of places in the continental US that had climate conditions similar to those where swallowwort is currently. This map is one of the projections I created for the potential distribution of black swallowwort. The black dots are current known locations, and the red area shows where the climatic conditions are similar to where the plant is found currently. This is only one of the projections I created. I came up with 2 or 3 for each species based on different parameters. This was a very rough and preliminary way of doing things, but it's a good start towards figuring out what places may be particularly threatened by these species, and working on the project taught me a great deal more about using GIS. In the fall, I will be attending graduate school at Cornell, where I will study crop and soil sciences, particularly weed ecology in relation to sustainable agriculture. This senior project gave me the opportunity to pull together and wrap up my experiences in conducting ecological research during my time at COA, expanded my abilities with GIS, scientific writing, and data analysis, and helped me prepare to conduct more research in the near future. Thank you.